Hi, everybody. Thanks for joining us at the 23andMe Parkinson's Disease Research Program webinar. We're going to give folks a couple of seconds here to join in the webinar, and then we'll get started. Just to let everyone know, if you have any questions during the presentation, please feel free to use the Q&A function to submit them. So that's down at the bottom. And also, we just wanted to let everyone know that this webinar will be recorded and we're going to make it available for future viewing. So we can share out a link to watch this webinar in the future. Great. So with that, we can go ahead and get started, and I'll start out with an introduction to our panelists. So next slide, please. My name's Helen Robotham. I'm a senior program manager for the PD research program, and I'm responsible for managing a number of our PD research projects. I'll be joined today by Daniela Coker. She's a scientist on our data collection team, and Daniela supports various Parkinson's disease research studies with survey development, implementation, and analysis. And Daniela will be walking through some results from PD-related surveys in the later half of the meeting. And then joining for the Q&A portion will be Eli Chanoff, who's our senior research associate. Eli is responsible for managing participant communication and the engineering aspects of our studies, and he'll be acting as a moderator for our Q&A session. And then Paul Cannon will be joining as well. He's the principal program manager of our Parkinson's disease research program. He joined 23andMe in 2014 and is responsible for leading the in-house research efforts and coordinating partner projects. He also brings a unique perspective to the program as a person who was diagnosed with Parkinson's in early 2011. So I've noticed we have some other folks that have joined. So just to let folks know, once you're in, if you have questions that you'd like to ask us, please submit them using the Q&A function, and we'll answer them towards the end of the meeting. We'll also try to answer some of the pre-submitted questions that folks sent us before the webinar. And just so everyone knows, this webinar is being recorded and will be made available for future viewing. Great, so let's get started. So today we're going to walk through the history of research at 23andMe and give some background information on Parkinson's disease, and then talk through how 23andMe can play a role in advancing PD research. Then Daniela will present some results from the exercise barrier survey, and then we'll finish it off with Q&A. So first, I'll start with some of the basics of how we conduct research at 23andMe. So when people become 23andMe customers, they can make an informed decision on whether to contribute their data to research. So our researchers always opt in, and customers who choose not to opt in do not give up any part of the 23andMe experience. Our research protocol and consent are approved by an external ethics review board, which is called an IRB, and our customers can cease participation at any time. The data that we use in our research is aggregated and de-identified, and no individual level data is shared without explicit re-consent. We make discoveries thanks to the participation of our customers. So we combine genetic data from surveys to perform research and fuel insights. And so now I'll talk a little bit about the 12 years of research of PD research at 23andMe. So we launched in 2009 with free or discounted kits to individuals with Parkinson's disease. And our initial goal was to recruit 10,000 participants. And we asked participants to take surveys about their experience with PD. We then started to branch out to or I should say before that, thanks to the contribution of our participants, we published our first paper in 2011. After that point, we started to branch out to sub-studies, starting with a skin biopsy collection project with the Parkinson's Institute and the Michael J. Fox Foundation in 2012. And in 2013, we reached our goal of 10,000 participants with PD, and the cohort has been growing since that point. We've continued other collaborations over the years. And in 2016, we entered a large collaboration with the Michael J. Fox Foundation. So this collaboration has included a medical records pilot, the Fox Insight study, and a microbiome study, among other aspects. In 2018, we launched the Parkinson's Impact Project, which is a study that I'll describe in greater detail later on in the presentation. And the Parkinson's Impact Project includes a number of activities such as a wearables pilot and a survey on exercise barriers, which Daniela will describe the results of today. Thanks to the contributions of our 30,000 participants with Parkinson's disease and the 196,000 individuals participating in our PD research program, which includes those who are at risk of PD or controls who have participated by taking a PD-related survey, we've been able to publish 21 publications, which is amazing and wouldn't be possible without the contributions of our participants. Great, so now I'll shift to talking a little bit more about Parkinson's disease. So although many of you who are on this webinar might already be familiar with Parkinson's disease, we wanted to provide some background information on the disease and how it relates to our current research strategy and ongoing projects. So Parkinson's disease is a progressive neurodegenerative disease that's related to the loss of dopaminergic neurons in specific regions of the brainstem. It's the second most common neurodegenerative disease after Alzheimer's disease. And in fact, the number of people with PD more than doubled between 1990 and 2015, and is projected to double again by 2040, as you can see in this figure to the right here. Parkinson's is defined and diagnosed on the basis of what we call canonical motor symptoms. So people are diagnosed with Parkinson's if is defined by the presence of bradykinesia, which is slowness of movement, combined with either tremor, rigidity, or both. So I mentioned that PD was diagnosed on the presence of motor symptoms, but there's also non-motor symptoms that are associated with the progression of Parkinson's disease, as well as non-canonical motor symptoms that are also associated with the progression of Parkinson's disease. And as this chart demonstrates here, we now know that the disease process begins years before an individual is actually diagnosed. And we call this period of time before someone is diagnosed, but when the disease process has started, as the prodromal phase. And this phase is associated with non-motor symptoms, such as constipation or loss of smell. It's also associated with significant neuron loss. And then the time at which someone gets diagnosed might not actually be reflective of what stage they're at in the disease process. So someone's diagnosis could be impacted by a number of factors, including access to care and the presentation of their symptoms. Right now, the largest unmet need in the treatment of Parkinson's disease is effective disease-modifying therapies. So while there are dopamine treatments that can be effective at treating motor symptoms, they do not slow disease progression. And in order to prevent Parkinson's disease, we'll have to intervene early in the disease process. And then in order to intervene early, we'll need to improve our ability to be able to detect and monitor prodromal PD. So how does 23andMe plan to help address these unmet needs? So we have a unique approach to advancing PD research. We know that traditional medicine is typically blind to the prodrome or early disease phases for most disease. So it's typically limited to treating or researching individuals who have been diagnosed, which is really the tip of the iceberg. However, thanks to the contributions of genetic and phenotypic data from our research participants, we can engage individuals who are in prodromal phases of the disease or early stages of the disease, or even earlier than that. And our model provides an opportunity to potentially prevent or delay progression to diagnosis. So that leads to the main goals of our program, which are to support research that can enable early detection and monitoring of prodromal PD, and to support the discovery and development of disease-modifying therapies. So one way we achieve those goals is through novel and deep phenotyping of cohorts that have an increased lifetime risk of PD. So in this example I've outlined here, we have two groups of people. One group has a typical lifetime risk of PD. We call that the control cohort. And then we have a cohort that we know is at increased risk of PD. And those individuals in pink are the little people in pink are individuals who will develop PD in their lifetime. So if we match these cohorts for age and other demographics, demographics, we can assume that the cohort with increased risk will have a higher proportion of people who will develop PD, and a higher proportion of individuals who are in early stages of the disease compared to the control cohort. Therefore, the phenotypes that are enriched in the risk cohort compared to the controls might represent early warning signs of PD. So for example, if both groups reported whether they experienced phenotype A and B, and then if the proportion of people experiencing phenotype A was the same in both groups, however, the proportion of people experiencing phenotype B was higher in the risk cohort, it would then be possible that phenotype B is an early marker of Parkinson's disease. So this is the principle that we're using for the Parkinson's Impact Project. So now I'll talk a little bit more about the Parkinson's Impact Project. So the G2019S variant in the LRRK2 gene is associated with significantly increased risk of developing Parkinson's disease. In the general population, about 1 to 2% of people will develop PD in their lifetime. And then in individuals who have one copy for the G2019S variant, the lifetime risk estimate is around 24%, although it varies for different populations. In 2018, we launched a series of research and engagement materials aimed at G2019S carriers, both with and without PD, to help us understand more about the potential early signs of PD. And we have a little over 4,000 people who are eligible to participate in the Parkinson's Impact Project. And the participation of these individuals is incredibly valuable to PD research, as they can offer key insights on disease prevention. So this is an overview of the ongoing activities that are part of our Parkinson's Impact Project, which we also call PIP. We launched with a series of cross-sectional and longitudinal surveys that we asked participants to complete every 3 months. Then we branched out to include a telemedicine collaboration with the University of Rochester, which is called Valor PD. We also recently completed data collection for a wearables pilot to understand how to operationalize a wearables study at 23andMe. And what we'll talk about today is some of the pre-work that we're doing with Radboud UMC to learn how to deploy an app-based exercise intervention. So to talk more about exercise, PD, and what we'll learn, I'll pass it over to Daniela. Take it away.
Great, thank you. So as Helen mentioned, so in this section, I'll be talking about the results from a survey that we fielded to better understand exercise knowledge, attitudes, and barriers of LRRK2 G2019S carriers in our PIP cohort. So for a little bit of background, aerobic exercise is recommended as a symptomatic treatment for people diagnosed with Parkinson's disease. And there's emerging evidence that exercise interventions, particularly those based on moderate to high intensity aerobic exercise, may help slow disease progression. And this is an area of ongoing research. And us at 23andMe, we're interested in investigating exercise as a preventative measure in populations that are at increased risk of developing PD. So to that end, we've been collaborating with experts at Radboud University Medical Center to deploy an app-based exercise intervention. So before we could start this app-based exercise intervention, we wanted to understand the attitudes and barriers to exercise in a population that is at increased risk for developing PD. So this is why we deployed our exercise barriers survey. And a couple guiding questions that we were particularly interested for this analysis were first, are individuals with at least one copy of the G2019S variant aware of the potential benefits of exercise for Parkinson's? And secondly, are individuals with at least one copy of this variant able to exercise as much as they would like? And if not, why not? What are their barriers to exercise? And on the flip side, then what are their motivators for exercise? So first, we compared key demographic variables between carriers with PD and those without. In total, we had about 500 survey completions between the time of data collection and when this analysis was done. And in general, both groups seem to be relatively well balanced. However, those without Parkinson's were less likely to have completed the survey and tended to be younger and less often married than those with PD. Next, we looked at the distribution of self-reported current physical fitness by PD status. And what we saw was that there was a larger portion of carriers without PD who rated themselves as having good or very good physical fitness. Whereas among those with PD, responses tended to skew a bit more towards moderate levels of physical fitness. And even when we restricted to people 65 years and older, we see a similar pattern between self-reported fitness and PD status. Next, we also asked survey respondents whether they were aware that exercise may help slow the development and reduce symptoms of Parkinson's disease. And because we thought that having a family history of PD may influence this response, we pulled that out as an additional variable to stratify on. And in general, what we saw was that most people were aware of the beneficial relationship between exercise and Parkinson's disease. This level of awareness was higher among those already diagnosed with PD. And among those without PD, having a family history did increase this level of awareness. When asked, do you have barriers that stop you from exercising as much as you would like? 57% of respondents without PD and 72% of those with PD said yes, that they do have barriers to exercise. And among those who do report having barriers to exercise, a large majority of people, regardless of PD status, said that they would exercise more with fewer barriers. For those who reported having any barriers to exercise, we then presented a list of 13 barriers and asked respondents to rate how closely each barrier applied to them. And because we knew that our group of PD carriers were on average a bit older than our non-PD carriers, we looked at barriers by both age group and PD status. So there are a couple of patterns I wanted to highlight here. First is that many of the same barriers appeared as top barriers for all four groups. So first was don't have the willpower or motivation to exercise. I have an injury or disability that makes it difficult to exercise. And I don't have the energy to exercise. If we look at the younger group of people for a moment, and that's defined by those younger than 65 years of age, for those without PD, not having the motivation to exercise was more of an exercise barrier for them, as well as not having the time to exercise. For people with PD, one of the top barriers was having an injury or disability that made exercise challenging. And also, half of the younger respondents with PD reported not having people to exercise with was a barrier, which was interesting because it didn't even make the list of top barriers for those without PD. As for the older group of folks, having an injury or disability that made exercise challenging was more of a barrier for those with Parkinson's compared to those without. And other than that, the remaining top barriers were pretty similar between the older PD and non-PD respondents. Not wanting to injure yourself was a top barrier for those without, and similar not wanting to fall was a top barrier for those with. Next, we also presented a list of 10 motivators for exercise and similarly asked respondents to rate how closely each motivator applied to them. So top motivators for all groups included that exercise improves the way my body functions, exercise improves the way my body looks, exercise improves my mental health, and exercise may help slow the development and reduce symptoms of Parkinson's disease. Again, just looking at that younger group for a moment, The exercise as a way to slow the development and reduce symptoms of Parkinson's disease was much more of a motivator for those already diagnosed. And those without PD had improved body appearance as a top motivator, whereas those with PD said being advised by a healthcare provider to exercise was a top motivator. And then on the other side, among respondents 65 years and older, a similarly high proportion of people, regardless of PD status, agreed that exercise to slow the development and reduce symptoms of PD was a motivator for exercise. A motivator that was more distinct to this PD group was being advised by a healthcare provider to exercise. So we have a few key takeaways from the results of this survey. First was that most LRRK2 G2019S carriers are aware that exercise may help slow the development of and reduce symptoms of Parkinson's disease. However, there's still a large portion that are not aware of this benefit of exercise for PD, so which indicates that there is room for improvement. And however, despite this higher level of awareness, more than half of survey respondents experienced barriers to exercise. Most of the top barriers and top motivators were the same between the two main comparison groups. And again, that's those diagnosed with PD and those without. In general, top barriers to exercise were not having energy or motivation to exercise. Having an injury or disability that made exercise challenging was more of a distinct barrier for those with Parkinson's disease, as well as not having people to exercise with, and that's particular for the younger folks with PD. And lastly, in general, top motivators for exercise were that exercise improves the way my body functions and or looks, and improves mental health. Among those already with PD, using exercise as a way to slow development or reduce symptoms of PD was particularly motivating, especially among the younger group. And being advised to exercise by a healthcare provider was also a distinct motivator for those with Parkinson's. So as for next steps for this work, we are currently in the process of writing up these findings alongside Radboud for publication. So stay tuned.
Great, so I'll just summarize what we've talked about, and then we can move on to the Q&A section. So in summary, our PD research strategy continues to evolve from the active growth of the PD cohort in our early years to an expansion to genetic risk cohorts. And now we're focusing on longitudinal phenotyping of prodromal and early PD, looking at novel phenotyping and risk prediction tools, and then piloting interventions in at-risk and prodromal cohorts. And then if we could move to the next slide. Great, and this I think is the most important slide. This research would not be possible without the 23andMe research participants who have participated in our PD research program, and in particular, those who completed the exercise barrier survey. So we just wanna say thank you to that group. We really appreciate your contribution to our program. We also want to thank the other members of the 23andMe Parkinson's Disease Research Program team, and then also Radboud University Medical Center, who is collaborating with us for this research. Great, so now we'll move to the Q&A section. So we'll just go ahead and have our panelists turn their videos on so we can answer some of your questions.
All right, thank you so much, Daniela and Helen, for that presentation. And thank you everyone else for being here. We really appreciate your interest. I'm gonna start off our Q&A with a few of our pre-submitted questions. And if you have a question that you'd like us to answer, please do submit it through the Q&A feature at the bottom of your Zoom screen, and we'll do our best to provide an answer. So I'm gonna get started with, what have you been able to discover using the genetic information of 23andMe users that you would not have found otherwise?
Great. Yeah, I can take this one. I love this question. It kind of feels like I submitted this question because it's so perfect. There are quite a few things that we've been able to do that we wouldn't have been able to do without the genetic information that our research participants have contributed. So there's about 90 variants that are associated with Parkinson's disease, and that's from the most recent large genome-wide association study that's been published. And those 90 variants explain about 16 to 36% of the heritable risk of Parkinson's disease, depending on prevalence. We include data from 23andMe participants is in that publication and has contributed to other publications that were the first to identify at least 38 risk signals that are associated with Parkinson's disease. So that's, if you think there's 90, 38 included were discovered from publications that use 23andMe data. That's pretty impactful and very, very cool. So the other group I would say is that our 23andMe customers are part of one of the largest LRRK2 G2019S cohorts in the world, to our knowledge. And the potential for insights coming out of the research that we're doing with that group are pretty substantial. So stay tuned. There'll definitely be a lot more from us in the future around that.
Cool. Thank you, Helen. I'm going to do one more of our pre-submitted and then move on into some of the ones that you guys are asking right now. What trend of Parkinson's disease is considered hereditary?
Yeah, so this is a big one. And I can start out with an answer, and then I think Paul can also jump in if he has anything else to add to this. So in general, and for PD specifically, there are some extremely rare genetic variants that are highly impactful, while there are genetic variants that are much more common that are associated with a much smaller increase of the risk of disease development. So what we call monogenic forms of PD, so meaning that the disease is driven by one gene, represent the minority of all forms of PD. So most people who have Parkinson's disease, it's not caused by one gene, and the heritability of Parkinson's disease is only explained by about 16 to 30% of genetics. So most interest in the PD space is focused on a couple of different mutations, a couple of key different mutations. So there's, I don't know if it makes sense to list all the genes here, but there's synuclein, LRRK2, Parkin, PINK1, and GBA that are kind of the main genes that people look at when they talk about PD. And each of those mutations and each of those genes kind of represent a different flavor of Parkinson's disease. So some features can have an earlier age of disease onset and faster progression of motor signs and more rapid decline. And then other mutations are associated with like slightly different phenotypes for PD. I would say one of the things we're interested in looking at is what does LRRK2 G2019S PD look like compared to PD that's not caused by LRRK2 G2019S. So there's some evidence that there might be some differences there, but it's definitely not conclusive. So we want to learn more about that. Anything?
Thanks. That was
Yeah,
good.
anything
No, I think those are living with the disease, obviously, it varies a lot. Different people, have same genetics underlying it. How you experience the disease can be completely different. And often that's to do with other things, compensatory mechanisms. If you're a great athlete, you're probably not going to fall over as early as somebody perhaps is a couch potato with the same underlying mutation. And as Helen said, I think that the G2019S is interesting. It's definitely a different flavor of Parkinson's. Sex ratio is clearly different. And if you have it, it's one to have because it's actually a slower progressing form of the disease. It's generally mild. So though if you're carrying G2019S, you're at much higher risk of getting PD over your life. That PD will generally be milder compared to, say, somebody who has a GBA mutation or someone like myself who has no known family history or mutation. Mine's presumably mostly environmentally driven, which another aspect that Helen didn't talk about much today is the whole aspect of the environment, how that plays into the disease. And we're also looking at that research in Joe is much earlier on. And so today we're focused on genetics, but don't forget about the environment.
Great, thank you both. One question that was submitted live is exercise benefit only seen in aerobic exercise, or is it also seen in balance and hand-eye sports like golf, skiing, and tennis?
Yeah, so I think it's mostly associated with vigorous aerobic exercise. Is the main studies that have been done. That being said, I think generally the advice that physicians give to people with Parkinson's disease, or just broadly in general, it's like any exercise you can do is good. And so the best exercise option is one that you will do consistently. So, if vigorous exercise, if, running isn't your thing and golf is, it's better to do something instead of nothing.
Now, this is Paul. Most of the data so far have been in people with Parkinson's already.
Correct, yeah.
And the prevention, we don't know, maybe mild activity is better. It's unlikely to be the case, but given how difficult it is to exercise, especially during the pandemic, I think the more information we can give to people, the better.
Mm-hmm.
And they always say, do something you like and something you'll stick with. I think Helen's right. Any exercise is better than none. One of the things we're actually looking at with BASO is how can we motivate people to exercise? And are there things on the phones, apps, sort of virtual exercise buddies who can help you perhaps exercise better or more than I currently do, for example. So that's a good part of what we're looking at with BASO, not so much what the exercise will do, but how can we get people to exercise more. Tricks of making gamification, which is obviously a big trendy word in Silicon Valley, make it more interesting and sort of, some people are very competitive, maybe that for those people make it more of a competition, other people maybe more collaborative. As you build a team together, your team is what's measured. There's many different ways. I think we're just trying to look at how we motivate people. The science is all very good, but unless you change people's behavior, it's all a bit of a waste. And so people living with the disease and people who are looking after people with the disease, you have to help us to make sure we understand what motivates people and what barriers are there and how to overcome them best. And it's all doing together.
Great. We have another question specific to exercise, which is it that exercise improves every condition and not just PD?
Great, thanks, Eli. And hi, Claudia. Nice to see you on the webinar. I think like in general, exercise is recommended, not just for PD. I think that's one of the beauties of it as a potential intervention for the prevention of PD, because there's no downside outside of avoiding the risk of injury from exercise. Exercise is good for you. It helps to prevent a number of diseases, and the fact that it helps to potentially prevent disease, PD, and then is often recommended as a way to alleviate the symptoms of PD is just an added benefit.
Cool. I like this question. Any studies on who would benefit from DBS based on genetic markers?
Ooh, that's a good one. I don't know, Paul, do you have any info on that?
Personal experience, get it as soon as you can. If you're ready for it, if you're eligible for it, and the doctor says you're a good case. It's a bit scary. I went through it 3 years ago. Not the most pleasant thing in the world, but it works incredibly well. My symptoms basically went away for the first 6 months. They're back now, as you can tell. But I'm very low medication, from 16 centimeter down to about one to two tabs a day, depending on the particular day. It does work, and so far my stimulation level is very low. And every quarter I go in and they check me in, and maybe every other quarter it's adjusted up or down. And so definitely there aren't genetic indicators. Maybe an extreme genetic form of PD might be hard, but as far as I know, the LRRK2 and GBA respond equally well. In fact, those of you who know Ben Stetcher is one of the bloggers in the PD space. He's just had DBS and has written up about it. If you check him out, I think his blog's pretty well available. His experience to date, he's sort of going to document, and he's good because he actually will document it, because he's a rabid blogger, which is good. So we can learn through him. But generally, I'd recommend anybody who's eligible for DBS considers getting it. And you can also join research. When you do DBS, a lot of the times you're not eligible for regular research programs. There's a lot of research going to these closed loop systems, which is where the DBS adjusts itself based on how you respond to it. That's very much more experimental. A number of the groups at UCSF locally and Stanford are looking at, and others in the country as well. And those you can do as part of your DBS treatment as well. So check that out on the web if you need some information, or just ping me. Helen, do they have my email?
No, but if you do have questions or comments, you can share it to pd-help@23andme.com. We can answer up.
Yeah, I'll drop that in the chat as well. Next question is, how many cups of caffeinated coffee per day is considered beneficial?
Yeah, this is a good one. And I've definitely heard this one asked before. So and I guess we should have prefaced all of this by saying that none of us are medical professionals. So please follow the advice of your own physician. But there is some research that prudent, quote unquote, caffeine intake, so which they considered 1 to 4 cups a day in early life could reduce the risk of developing PD. And the effect is likely tied to caffeine as opposed to specifically coffee beans. So there aren't really definitive guidelines on caffeine intake or how much it could impact your individual risk. But I think if you're someone who enjoys drinking coffee and wants to have another excuse to, maybe have that extra cup, this is a good reason to.
So you said just pass up the full fat latte because of the milk and the fat.
Yes,
calories.
potentially.
All right, another question for you is, do all cases of PD involve one or more genetic variants? I had thought not.
Correct, yeah. So I think from what we understand about PD now, the PD genetics attribute you can attribute about like 16 to 36% of PD to genetics. So, the vast majority of that is not genetics. The other risk factors for PD, which we, as Paul alluded to, we haven't talked about too much here, environmental exposures like exposures to pesticides and other kinds of chemicals, and head injury is another huge risk for Parkinson's disease. So those are some of the other risk factors that are associated with Parkinson's.
And the biggest one's age. We can't do much about that. So, the alternative's not much better. And so I think with the exception of some very rare variants in PD, which are less than 1% of the whole total, it's really environmental and time. I mean, it's just injury accumulates, it's probably random in a lot of us. Bad luck, whatever. The good thing is it's manageable and we're working ways of stopping the progression and getting it earlier. Early in, we may be able to help people get away from things that are bad for them. So perhaps if you've got a genetic makeup or even a history of an earlier life exposure, then maybe you shouldn't be working in the chemical industry in a particular area. And we get some better information there. It's just a lot harder to prove that association than to say genetics, which is more obvious. So 23andMe is based on the genetics, but it's how people use the genetics to live better lives through managing risks. And that's much harder. And association is never causation. And there's lots and lots of spurious data out there. We're trying to filter it down, and over time, with your help, it's just focusing on the key factors that influence both people's risk of getting disease and once they've got the disease, how fast it progresses. Because even if for those of us that have PD already, it's not too late. Discoveries made where they have most impact in stopping PD starting will also help us, I think, if stop it. If my disease stopped progressing in the morning, that's great. I mean, I'm still getting older, so it's not be as good as it would be otherwise. Are we ever going to turn the clock back? I think people who think maybe stem cells will help, maybe, but it's a huge long shot. But in the short term, things that slow down the early part of the disease process will actually help, I think, the rest of us have a slower progression, therefore a longer life. Anyway, I'm probably waffling on too much. I'll let the others get back because they do much better of a job about talking about this now than I do.
Early in the presentation, you listed skin biopsy in relation to PD. Could you please explain?
Yeah, so this was actually a project we did well before I joined 23andMe. So we helped to recruit participants who were actually carriers of both a LRRK2 G2019S variant and a GBA N370S variant, which is rare to, it's quite rare to have both those variants. So we helped to recruit them for a clinical trial not a clinical trial, a study that the Parkinson's Institute and MJFF were conducting to use their skin cells to, I believe, create stem cell lines. I'm not sure, Paul, if you remember the exact details of that, but it was one of our early projects.
Not recommended. It's very painful. These are the old skin biopsies. If you've done research, involves like a full dermal punch. And so you think such a little cute little pile out of your skin, it's not very nice. Turns out there's one of these things, if you wait long enough, Roche have got some really interesting papers come out. They can show alpha-synuclein in the nerves in the skin by histochemistry. So the antibodies now better, so we can actually detect alpha-synuclein, which is one of the early components of the disease or thought to be misfolds. And it actually looks like it's higher in people with PD than people without PD. And so now 7 or 8 years later, we can actually do the experiments. So part of our trick is actually to wait until the technology is ready. As Helen said, we're doing pilots around she mentioned pilots around the wearables, we're doing pilots in microbiome and other things. So I think now the technology is getting close to being ready for prime time. We could have done it 10 years ago, but I don't know where it would be. The genetics has been ready for a long time. That hasn't really changed much. Just more and more people gives better, a finer lens, if you like, the way of focusing on the disease. But the underlying science hasn't really altered. But the other areas are really altering very fast. You have to keep an eye on them.
Thanks both. Are you expecting to find more genes that cause PD? Did I cut out for a sec?
Oh no, are you expecting to find more genes that cause PD? Is the question?
Right.
I don't know if we'll necessarily find any of these more like major variants that I referenced. So that's kind of 6-ish or so variants that are associated with a significant increase of PD. And again, those ones that are associated with a significant increase of PD tend to be more rare, while genetic variants that are associated with like very tiny increase of PD tend to be a lot more common within the population. So yeah, it's possible. We're definitely possible to find more than these 90 variants that explain some of the heritability of PD. But how valuable they are, because it's such a small twinge in risk level, is yet to be seen, I think.
Cool. Are there specific genes that point to young onset PD? What are triggers for early onset Parkinson's? What is the difference between early onset and PD that comes to older people? That was one question.
Yeah. So I can start with an answer on that one. So just in terms of terminology, we consider young onset PD to occur in people who are younger than 50 years of age. And the symptoms are similar to Parkinson's disease, but young onset PD is associated with a family history of Parkinson's disease, slower progression of Parkinson's disease, and then more side effects from dopaminergic medications, and more frequent dystonias, which is like cramping and abnormal postures. But the individual experience of someone with young onset PD can really vary. So just because someone has been diagnosed with young onset PD doesn't mean they necessarily experience those things that I just outlined. And I think one of the biggest differences, which I think people maybe don't necessarily consider in the way that an individual experiences PD, is that those who are getting diagnosed younger in life might be in different stages of their career. They might have different responsibilities around like children or parental responsibilities. So the way they experience the disease within their life could be really different from someone who is older or at a different stage in their life. So in general, genetics play a pretty large role in young onset PD. So I think, I'm not sure the correct way to say this, but if someone's diagnosed with young onset PD, it's more likely that it could be associated with genetics than someone who's diagnosed with PD that is not associated with genetics. And in terms of research that we're doing in that area, we're actually collaborating with Dr. Pilstrom at the University of Oslo to contribute data to a young onset Parkinson's disease genome-wide association. So we're still doing work in that space.
Can I quickly jump in on that one?
Yeah, please.
Yeah, so you're right. It's basically functionally derived on age. That's how we're going to change as diagnosis improves and people's awareness of disease improves. I think we'll have to move it sort of back a bit, because I mean, I don't know, I was diagnosed at 48, but I actually had disease symptoms for 6 years before that. Looking backwards, of course, at the time didn't know, but 42-year-old male who presents to the physician isn't really generally thought to have Parkinson's. So that's kind of the issue we're dealing with. But the other thing is, if you're extremely young, under 40 or under 30, it's always almost genetics. There's a particular set of mutations called Parkin and Pink1, if I get those right. And they're recessive, they're rare. But they always, if somebody has disease that early on, it's almost always those two genes. Good thing is you have to be homozygous for them. So if you have a child at that stage, it's very unlikely your partner also has the same mutation, and therefore the kids will be okay. But people worry about kids. So IOPD, again, if you have IOPD, the rate of family history is very, very low. It's like higher perhaps than in the general population. But I wouldn't be concerned. You have children who get tested if they have particular genes, but just because they don't have it doesn't mean they'll get Parkinson's. I think increased risk of having a family member, first to be relative, which is genetically related to you, it's only fourfold. It goes from about 1% to 5%, something in that range. And so really don't worry about it. Keep living your life, be healthy. And if you want to have kids, have kids. It's not like Huntington's or any of these other really nasty diseases where there's a much stronger genetic component to it. And people who are children of people with the disease know it's going to happen. I think most of us, it's not the case. And so if you want to have kids, don't feel bad about having kids. If you've had kids and you've got PD, don't worry about it either. The genetic contribution is relatively small, it gives insight into the process, but it doesn't really, it's not deterministic, if you like. So you get the gene doesn't mean you get the disease. Even people with very high risk, as Helen mentioned, it's only 30%. Well, 2 carriers get PD in their lifetime. That means more than half don't. And that's gene, we think, is a very strong influence. So it's important to realize it's not a death sentence. It's certainly not informative. It just tells you about risk, and you can manage it. If you know you're at risk, change your behaviors, exercise, etc. It doesn't do any harm, and it's all good. That's where the genetics is useful. It doesn't tell you what's going to happen to you, but it says the risk of this, and therefore you might want to change certain behaviors and avoid certain lifestyles. Preaching, I'm going to get off the soapbox now, but there you go.
Thank you, Paul. I'm going to move into another pre-submitted question. Are there new drugs in the pipeline?
Yes. So I can start with this one, and Paul, I'm sure you have more to add. Denali Therapeutics, I think, is the most potentially interesting one. They have a LRRK2 kinase inhibitor, which is potentially our best chance at a disease-modifying therapeutic. So they have planned to advance to late-stage development in 2021. They've publicly shared this information. And so that's an interesting one to keep an eye out for. There's also a couple of treatments that are based on GBA, the GBA gene. So one's being developed by Prevail Therapeutics, and then one by Sanofi. And then Roche also has a monoclonal antibody targeting alpha-synuclein that they are working on as well.
Cool. Are you looking for participants who have one or both of the test variants?
Oh, like LRRK. I'm assuming that means looking for people who have a LRRK2 G2019S variant and a GBA N370S variant. So that's how I'm going to interpret that question. Yes, is the short answer. So as most of the activities I outlined for the Parkinson's Impact Project have been directed towards people who have one copy of the LRRK2 G2019S variant, at least one copy of that. We haven't excluded folks who have a copy of the GBA N370S variant as part of that research. We would like to do more research in the future with individuals who have the GBA N370S variant without the LRRK2 G2019S variant. Similar, if I talked through our strategy, how we're interested in looking at cohorts who have an increased risk of Parkinson's disease because it can help us identify early signs of Parkinson's. So the GBA N370, people who carry the GBA N370S variant also have an increased risk of Parkinson's disease, although that increased risk is lower. But similarly, they are a group of people who could potentially offer a lot of insights into disease prevention. And I guess I should also, go ahead, Paul.
I interpreted you guys to do only people with both mutations, which of course is incredibly rare. But if you have both, do come forward, because they're fascinating about how they to interact. That's really just my scientific interest. Anybody who's got increased risk and you want to do research, we're here for you. Come on in, kind of thing. Fun to do the research. Even controls. I mean, just get into it. Learn about Parkinson's. We try and give these webinars to give feedback to participants, but also to controls and family members and stuff about what's going on with the science. I think there's other stuff you'd like to hear. Let us know. The PD help, is that the suggestions for topics they might want us to cover in future? Maybe the microbiome would be of interest to people. Don't know. So. Yeah,
and I should also say if you're interested in participating in various types of PD research, so, there's always the research stream within your 23andMe. If you log into your 23andMe account, there's various surveys that are available. We also email eligible participants with research opportunities when they're available. So, if you're part of Parkinson's Impact Project, you get an email from us every 3 months with a new survey to take. And that's how we tend to communicate. So if you're interested in studies, yeah, definitely keep an eye out on your email. Yeah.
Along those lines, will you be recruiting for participants in testing the exercise app? And if so, when will that start?
This is a great question. So yes, we will definitely recruit from our 23andMe research participants for that particular study. It's harder to say when that'll happen. So we're still in the very early stages of planning this out because there's a lot of moving pieces. So as you can imagine, it's difficult to coordinate with a couple different collaborators. And then, we're also going to be sending people to download an app and also potentially collect other types of information through their phones. So there's a lot of moving pieces. So it's going to take us a while to plan this. So I can't give a firm timeline, but I can say that we will definitely be emailing folks when that opportunity is available.
Just to reiterate my earlier point, I think it's worth waiting and do it right rather than rushing and doing it wrong.
Yeah, exactly.
Sometimes those who have the disease are a little impatient for things to happen, but on the research side, it just takes the time, unfortunately, sometimes. And I'm always better off just taking a breath and designing the study properly so we can tell what the answer is, rather than rushing into it and regretting 12 months and millions of dollars later that we did something wrong. Mm-hmm. And so I've been trying to preach patience here, which is very interesting because I'm normally the person pushing hard, but Anne is the CEO of 23andMe she wants everything to happen yesterday. And so it's always hard to slow her down, but she gets the science takes a while, and we're doing a really nice job. Helen and the team have actually put a good program together that I'm comfortable. As I exit actually through having PD, they'll be taking good care of us all. From a research perspective anyway, if not from a doctor's perspective.
Thanks, Paul.
All right, we've got a lot more questions. I'm gonna try to get through a few more, and then Helen, will you just cut me off whenever we should stop?
Yeah, sure.
Eli, we can always answer them sort of in the chat anyway, right? That's going to be, can we add to the transcript or the chat afterwards, or just do it as a separate email maybe?
Yeah, I'm not sure.
We can maybe lump together where there are themes coming out rather than Eli having to struggle through and pick them for us. There's a lot in the chat. Maybe we can look at them over the next few weeks or so and just get us an email blog. If someone has a theme to response, might be the easiest way to deal with it.
Yeah, we
don't get the question now. Most of the questions, there are some questions probably won't get to, like, what's 23andMe up to? Should I buy the swag? I mean, it's silly questions that people always send in these things, but most of the questions I've seen have been really good. And so we'll try to get answers to most of you. It may take a few weeks for us to just distill them down. If it's not exactly the wording of yours, probably been addressed, so just look for, you have to read all of them to see if yours is there. Anyway, does that make sense?
Yeah, yeah, we can definitely.
Too much now. We've got 10 minutes, I guess, right?
Themes that we didn't get to share out.
Yeah, that sounds good. Is there anything you guys wanted to end on?
It's 9 o'clock on the East Coast, and so we should be careful about people's time there as well.
Yeah, Eli, do you want to give us maybe 1 or 2 more questions, and then we can close out once we hit 6 p.m.?
Totally. How about starting with, are there any genes that prevent PD?
Ooh, that is a good question. Paul, do you know on this one?
The simple answer is, of course, the PRS area, in the multi-gene area, there's always 2 alleles. There's a risk allele and there's a non-risk allele. And so in theory, yeah, there are people who have none of the risk alleles, and their risk of PD is presumably less than on average. Do we know what they are and what they're telling us? Not so much. I mean, I think there are some things, let's see if it's right. That's a hard one, actually. It's much harder to show that genetically, and the statistics are harder. That's one we should probably get back to, because I think there's an answer there, but Carl, we need our StatGen, who's not with us today, is stuck in Canada, I think, in the heat wave in Western Canada, but maybe we can get Carl to opine on that one for us.
Yeah, definitely. We can follow up on that one. Mm-hmm. Yeah, it's easier to see. It's easier to see something that causes something than something that's protective. It's a little tricky.
For the absence of something, which is always hard, it's a negative result.
Yeah, exactly.
Cool. All right.
I'm gonna
finish off with one more question, which I think we could spend a whole hour answering, but I'm gonna limit us to 3 or 4 minutes, which is, what is the future for 23andMe Parkinson research?
Great. Yeah, I did not submit this question, even though it sounds like one I would have a plant in the audience to ask. Yeah, so happy you guys asked about that. So as we talked about, the exercise intervention is a project that we're really enthusiastic about, and definitely one of the big things that we're looking forward to in the next year or so. So that's one piece of work. We're also working on analyzing the data from our wearables pilot. So we hope to have some information to share about that. Because it's a pilot, it's a very small amount of people who contributed data to that. So I don't know if we'll have any really interesting insights coming out of that, but it will help us to understand how to operationalize a wearable study at 23andMe if we choose to do that. So those are like two of the studies that were coming up. Other things that we want to move into, so we really are focusing on novel and deep phenotyping of at-risk cohorts and prodromal and early PD, and then also other stages of PD. So we are just wrapping up a microbiome study that we did with individuals with PD. And we really want to start moving out also into mobile data collection. So, we ask people to contribute surveys quite often, but there's some really cool tools out there for monitoring symptoms of PD and then also early signs of PD. And we'd love to partner with different groups who have access to those tools and then use them within our research community. So there's a lot of stuff that we want to do. It's just a question of picking like which are the interesting opportunities that we want to really move forward with. So stay tuned. And I should say before we close out, we really appreciate that you guys came to watch this webinar and blown away by the number of questions. We really appreciate your guys' engagement with this group. We'll be sending out an email with a link to a survey afterwards to ask you basically whether you liked a webinar, whether you want to see more of these in the future. So if this was something that you enjoyed and want to see more of, please take that survey to let us know. Or, if you would like to see less of it, you're welcome to say that as well. So yeah, thank you all for joining, and thank you to all the panelists for presenting as well. And I think with that, we'll close out and say goodbye. So thanks all.
Thank you, Helen. Thank you, Eli. And thank you, Maddie and Daniela.
Great. Thanks, guys.
Thank you.